Well, hello. Hopefully there's some people out there. I can't tell yet, but I'm so excited to be here today because I get to do this Instagram Live. It's been a while since I've done Instagram Lives, and I've gotten a ton of questions. And, you know, I was commenting earlier to one of our people here, amazing questions, like really good questions that require answering and sometimes aren't so obvious. So I really thank you for that. So as people are rolling in, I want to talk about a little bit about myself. I'm Dr. Mark Pimentel. I'm Executive Director of the MAST Program here at Cedars-Sinai. We are really trying to figure out the causes and treatments for SIBO and IBS and EMO and all these different chronic GI conditions. And to that extent, we have really come a long way in the last few years, especially in the last 2 or 3 years, and I'm going to hope to cover some of that. But for example, we have a new trial for a drug that we think works better than anything previously for IBS-D on the basis of even hydrogen sulfide. It really is good for hydrogen sulfide. And then a new drug that can maybe reduce methane by up to 97%. Can't use it in humans yet, but we're coming along. So super excited. And hopefully we'll unpack some of this with you today. But now that some of you have rolled in, I just have a question. If you could, all I know is about throwing a heart or a thumbs up, I guess. But how many of you have had a breath test already and have questions about the breath test? Can you just throw a heart? Well, nobody. Nobody, or everybody's still getting in. Second is, how many of you have IBS with diarrhea? Not that accessible in Canada for breath testing. Okay, so we're seeing some people from different places and some people with IBS. And so what I'm going to try to do is I'm not going to do a lot of talking with slides or anything like I sometimes do. What I want to do is I want to try to answer the questions. I've gotten questions ahead of time. I'm going to try and follow the questions as they come through on the live so that I can answer some of your questions as well. But so some of you have had many breath tests. I just saw Linda Hall. Thank you so much for responding to that. So there's a list of EMO, SIBO, and breath test questions. But please send your questions along, and maybe they'll synchronize with some of these questions that were sent ahead of time. But so there's intestinal methanogen overgrowth. So for those of you who don't know what these things are, intestinal methanogen overgrowth means that there's an organism in the gut that produces methane. It's not a bacteria, it's an archaea, even older than bacteria themselves. And the bug that produces methane is Methanobrevibacter smithii. So Methanobrevibacter smithii, when it increases, you get constipation. And so that's one of the topics that we're going to cover today. And so when you eat some of the bacteria, some of the food you eat, especially if it's non-digestible products, you get the bacteria get exposed to this food products, and then they produce gas and bloating and distension. Normally, you shouldn't have this excessive bacteria. You shouldn't have these types of bacteria and these archaea in your small intestine and your colon in this elevated capacity. And we call these sort of situations microbial overgrowth syndromes. So now we have the EMO, which is methanogen overgrowth, which is too much methane, constipation. Thanks, Linda Hall, for saying you have constipation for 9 years. That's a long time. We're going to try and see what we can do to answer some of your questions. And then there's SIBO, which is small intestinal bacterial overgrowth, which are the Ferraris of fermentation, which is E. coli and Klebsiella. And then there's the new one, which is intestinal sulfide overproduction, which has really become important because intestinal sulfide overproduction is the most toxic thing to your intestine. When we study the small intestinal wall, we see that the most dramatic changes you really hate when you have hydrogen sulfide in the gut, and that causes more severe diarrhea, more severe symptoms in general. So let's start with some questions. So before I get to that, I do know, and I see some of the comments that are coming across, that a lot of the physicians in the community really haven't gotten caught up. One of the problems with breath testing, even though it's been around a long time, modern breath testing, especially 3-gas breath testing, has really come only to the front in the last 3 or 4 years. So we're really starting to see a better understanding using this breath test, but sometimes filtering that or getting that down to the primary care, even the primary care GI level, takes time and education. And there's a comment about hydrogen sulfide in Europe. There is actually Colab.com. Colab in Europe, which is based out of the UK, can do 3-gas breath testing, if you go to their website, anywhere in Europe and Australia. So that's a new development that I didn't even know about a week ago. So that's really exciting for those of you in Europe. But somebody asked a question, so let's get to those. What about acids? Can acids contribute to intestinal methanogen overgrowth? It's a little tricky answer, but basically the point of this question, and obviously somebody's heard me speak before on this, is that anything that produces hydrogen or gives away hydrogen can be a source of hydrogen for the development or production of methane gas, which then leads to constipation. So if you have too much acid, you will get more methane. But not a lot of people have too much acid. It's mostly that if you can get acid down or eat less acid, so less maybe vinaigrettes, less, and not apple cider vinegar. You wouldn't want to do that with intestinal methanogen overgrowth because it will increase methane more. So yes, acid can make methane more. So there's somebody says Colab is very expensive, sadly, but that at least is an option. So thanks for that comment. Ruben13 says, my internist thinks SIBO emo is controversial diagnosis, and she gets that from the common online references doctors use. This needs to be corrected. Let me address that before I go back to the questions that were pre-submitted, because I would lose it in real time in a few seconds. I don't understand that. Maybe you can share with them that there is a North American consensus on breath testing, which encompasses all of the North American experts. That was in 2018. 2020, there was a European consensus on breath testing. There's an Asia, Australia consensus on breath testing, the Brazilian consensus, and 2 weeks ago, the Argentinian consensus. And they all tout the merits of breath testing and the benefits of breath testing. So this is a consensus. This is not just me talking about breath testing. The world does breath testing. So it's ironic that these primary care physicians still don't get it yet. So another question that's come in is, Dr. Pimentel had mentioned PPIs do not cause SIBO emo. Can you explain further? So one of the things about small intestinal bacterial overgrowth is that there was this notion that if you had low acid in your stomach, that could lead to SIBO. So the answer to that is, it's true if you have zero acid all the time in your stomach, and this only happens in extreme situations for autoimmune disease of the stomach, which is atrophic gastritis, for example. You have no acid, and maybe that produces a little bit of SIBO. But if you just take a PPI, it's not 100% coverage, and so you get some acid exposure, and you only need 5 minutes of acid for it to kill bacteria. So I think that's the reason why PPIs don't cause acid, or don't cause SIBO. And I just got to shout out, Brazil loves you. I love Brazil too. So hey to those from Brazil, welcome. Nice to see you here. How do you manage GERD and SIBO at the same time? So that's a dovetail with the other questions. So you don't want to avoid the PPIs. You have to manage the GERD. But what I often do for my patients who come in, let's say they come in fresh, never seen a physician before, they have SIBO and GERD, I'll treat the SIBO first. Because GERD is both the valve being low in pressure, the valve being between your stomach and your esophagus, that can sort of give you a risk factor for GERD, and that the pressure below the diaphragm, meaning the abdominal pressure, is higher than the pressure above the diaphragm. So if you have a lot of bloating and distension, the pressure below the diaphragm is higher than the pressure above the diaphragm. So let's get rid of the SIBO first, reduce the bloating, and if the GERD goes away, hey, you don't need to take a PPI. So that's what I would do. And then Rubin 13 says, what about EMO and GERD? Same story, but EMO's a little bit more complicated because EMO slows your gut down. So if you slow your gut down, you also get that back pressure from EMO. But the answer is the same. Treat the EMO first, and if the EMO's gone and the GERD goes away, then you don't need a PPI. That's how I would do it. But there's one more nuance to that. We published an abstract at DDW last year where we found that hydrogen sulfide, which is that third gas, when it's up, it's associated with GERD, and hydrogen sulfide can lower directly the pressure of the connection between the esophagus and the food pipe. And so hydrogen sulfide may be a whole different issue in a cause of gastroesophageal reflux disease. Somebody asked here, what are the links between SIBO and SIFO? So small intestinal fungal overgrowth means fungus is growing too much in your small intestine. It's almost always candida. So when candida is elevated, it's usually for the same reason that SIBO is elevated. And so in a study that we presented again at one of the big GI meetings, we noted that SIFO does occur, but it occurs much less commonly than SIBO. But when you have SIFO, small intestinal fungal overgrowth, you almost always also have SIBO. So you've got to kind of treat both. But in a patient where you take rifaximin, you don't quite get better, you might want to try a treatment for SIFO. The problem with SIFO is there's no breath test. There's no breath test for SIFO. You have to actually go in and culture and get culture of the organisms. Somebody just said there's so much pressure with EMO. Absolutely, EMO. So nobody wants to make this decision, but if I'm in clinic, the patients with constipation and EMO are often more miserable than the patients with diarrhea and SIBO. They're both miserable. Please don't get me wrong. What I'm trying to say is that when you have the miserable sensation of constipation and bloating and you never go to the bathroom, you never get relief. But when you actually go to the bathroom because of the diarrhea, in the diarrhea patients, at least for the moment, you get relief. And so, anyways, that's a really they are different, and EMO can sometimes be more traumatic. Lucy Bender asks, are Sjogren's patients more susceptible to SIBO or EMO due to the lack of saliva? I'm desperately searching for my root cause. I saw other questions about antivinculin. So let me take the Sjogren's and the antivinculin in a slightly more advanced direction and then come back to vinculin again. Antivinculin antibodies are caused by food poisoning. So what starts this whole thing for most people is they get food poisoning, they develop anti-CDTB antibodies because that's the antibody to the toxin in food poisoning. But that antibody looks, or the toxin looks a little bit like you, the protein vinculin, and then you start to form antibodies against yourself. And then the movements of the gut diminish. Antivinculin antibodies are found in two conditions, IBS with diarrhea and scleroderma / possibly Sjogren's, because they often go together, scleroderma and Sjogren's. So if you have Sjogren's, it might be worth measuring antivinculin antibodies because they could be elevated, and that may mean your gut is slow, and if your gut is slow, you could have SIBO. And so antivinculin antibodies are linked in that way. But the other part about antivinculin antibodies that answers a question that I saw coming through earlier is that the higher the antivinculin antibodies you have, and the higher the anti-CDTB, the more SIBO you have, and because it's a greater neuropathy, the higher the vinculin antibodies are, the more difficult your SIBO is to treat, which means you may have to take rifaximin more times a year than other people, or you may have to be more aggressive in managing your symptoms. Now, somebody asks here, could food poisoning cause EMO? The answer is yes, we do see that. I had a patient just this morning I was talking to on the phone from Europe, and he has emo and had food poisoning. And so, and although it happens less often, so if you were to stratify this, the SIBO patients or ESO patients is about 56% are positive. IBSD is the same group. And so it's positive there quite commonly. In the IBS mixed group, it's about 40%. And in the IBS constipated group, which is mostly the emo, it's about 25%. But healthies are less than 10. So yes, it's there, but it's less common. Are there effective treatments to reduce antivinculin antibodies? The answer is there are two treatments that can do it, but they're very intense. And we would only use this, and we have used this in patients who are extreme bloating and extremely high vinculin, and that is IVIG, and we have to coordinate that with a neurologist or a rheumatologist because this is an autoimmune neuropathy. So we use it under that inference or under that diagnosis. Plasmapheresis we have done for patients who are in and out of the hospital with the kind of bloating that you would never imagine, never hope you have. And that works. That will make them better. So it's, but we're trying to discover, and we've come a long way. We actually know the antibodies. We know the sequence of the antibodies. We know the B cells that produce it because we're marching towards the development of drug that could reduce those antibodies. So you have to stay tuned for that. We are close. We're getting there. We don't have a drug for that yet, but that is the mission. And, it's a very expensive mission, but we're working towards that. And we hope to get there as soon as we can. I had some food poisoning and I tested positive for emo. Yes, so that can happen. So the only way to know is to do the anti-CDTB, anti-vinculin test. D. Tennant asks, can you have elevated vinculin with IBSD and not have SIBO? So you have to explain to me what you mean by not have SIBO. So a couple of problems that we see out there, and there's a beautiful paper we just got published that kind of answers this. If you are looking for SIBO with a glucose breath test, you're going to miss it because glucose undercalls SIBO. So if you did a glucose breath test and it's negative, I'm not believing you don't have SIBO till you do lactulose. So that new paper says, period, glucose is missing patients with positive breath tests that could have been positive if you'd done lactulose. So you probably need to do that to be sure. The second is it could be ESO, intestinal sulfide overproduction, because you could have one or the other or both. So unless you do the 3-gas breath test, I'm not sure if you have SIBO or ESO or emo or none of those. So you have to kind of do all of that. Sarushka, 25, please provide an update on FDA review of the new methane reducing drug. So where we are now with that is we can get over 90% reduction in methane in the lab. We've done some animal studies on this. We're now progressing through the safety studies that are required for the FDA. And then documents will be submitted to the FDA sometime probably early 2027 with the hopes of being able to do phase 1 trials in humans in 2027. That's where we stand currently. The Pixie journals just wrote high hydrogen levels that don't lower with rifaximin. Some of these questions are very similar to the ones I have that were pre-submitted. So I'm trying to go back and forth. If your hydrogen is super, super high, often those patients are harder to get down with rifaximin. And we see that in our clinic. We've seen that for 20 years. It'll come down, but it won't come down enough. And so I point those patients in the direction of elemental diet if I can't get the hydrogen down. An elemental diet usually does the trick. 2 weeks of an elemental diet. If you don't know what that means, because you've not heard of that, it's basically all the food's broken down. It's a product. You can either use Vivanex or the new one, which is much more palatable, is M-Biota. It's a elemental diet that if you take it for 2 weeks, about 70 to 80% of patients, the hydrogen is gone, the methane is gone. So that can be very, very useful. Sarushka asks, can we enter clinical trials? Oh, and an update on the drug for IBSD. So those go together. So we are the new drug for IBS-D slash hydrogen sulfide is in trials now. We just had our first few patients running through and we're enrolling for it. So if you, and this is the rifaximin and NAC, which TXMKV just pointed out, this is enrolling now. So if you want to enroll in that trial, you have to be IBS with diarrhea, and you have to have had a colonoscopy, negative colonoscopy, in the last 3 years. Those are the two big, there's other exclusions, but those are the two big ones. So I'm going to give you a telephone number if you want to participate. You have to be in LA because it's 6 visits, so it's hard for people who are outside of LA. But it's 310-423-0617. We can send it out to you as a separate, but that's our research number. That's 310-423-0617. So that's for IBS-D patients. That trial's going. We're getting 225 patients, 3 centers around the United States. If you live in Connecticut, Connecticut Gastroenterology is going to be doing that trial, and then there's going to be a third site in South Dakota. So we can keep you posted on that. Okay. In terms of breath testing, a question that's come in that's really important for today is, does GLP-1 usage impact breath testing? And so GLP-1 usage has a tremendous effect on breath testing, because if you're on it, your breath test is going to be 0. You're not going to have a positive breath test. I'm not going to be able to see any rise. And so it's almost useless to do a breath test if you're currently on GLP-1s. You can't rely on a negative test. So that's another reason why earlier somebody said, I have a negative test. I'm not saying that person was on GLP-1s. I don't know them. But if somebody was on GLP-1s, that could lead to a false negative test as well. Somebody asked about M-biota causing diarrhea. It's a liquid diet, so midway through the M-biota, almost everybody has a touch of diarrhea because it's just liquid going through your system. That is usually normal, and so it's not a concern, and maybe a sign that things are working well, actually, because if you started with EMO, having a little bit of diarrhea means things are opening up. Hypothyroidism and constipation. So this was a question that's come across as I'm going through all these questions. Hypothyroidism definitely is linked to constipation, although I don't see it that often. So if you look in the textbooks, if you're hypothyroidic, you could have constipation, but most people are treated for their hypothyroidism, so that's not going to be causing constipation currently. But Dr. Mathur from our group has really done a great study looking at the relationship between hypothyroidism and SIBO. And just the fact of having an underlying hypothyroidism appears to be associated with SIBO, and replacing the thyroid may reduce the amount of SIBO. So I encourage you to look for that study that's been published, and it was very well received. Excellent study. What about antivinculin? So do you recommend retesting antivinculin antibodies? So antivinculin antibodies, once they've established, they take a long time to go down. Like we're talking years. But what I do is I measure them yearly to see if they're going up or they're going down. If they're trending down, eventually I can get people off their medications or treat them less. And so I have some patients where over years the anti-CDTB or antivinculin went to normal, and then I could use less prokinetics. So prokinetics, I mentioned prokinetics, maybe this is confusing. So one of the things we do with people with high vinculin at our program and SIBO is we know that the vinculin antibodies are causing the SIBO. We treat the SIBO, and then we often put people on a prokinetic at night to make the cleaning waves of the gut go more, because the antivinculin reduces those cleaning waves. So we give a medication to get those cleaning waves going. And by doing that, you use less antibiotics, less rifaximin, and less often. And then what happens is, if the vinculin antibodies over a couple of years start to come down, you might be able to get off the prokinetic. Maybe you don't need it anymore. Maybe the cleaning waves have recovered. That will happen if the vinculin goes back to normal. Okay, let me go to another one here. Are there ESO food triggers? So ESO is intestinal sulfide. I'm just going to read this out loud. I have antivinculin, but been SIBO free for 3 years. Should I lower? I'm losing it. Should I lower my prucalopride? Generally speaking, if I have a patient who was positive for antivinculin and the antivinculin starts to go down or goes to normal, I start to wean them off the prokinetics. That's what I do in my practice. So ESO, intestinal sulfide overproduction. This one is the sulfur utilizing bacteria. So they can use sulfur from amino acids like cysteine, homocysteine, or they can use sulfur from just sulfur-based anything. And what they can do is produce hydrogen. What they do is produce hydrogen sulfide. And so the ones that produce hydrogen sulfide from sulfur is usually sulfate-reducing bacteria, and then the other organisms produce it using the amino acids and other pathways. But the bottom line is they produce hydrogen sulfide. So we've argued for a while that maybe if you go on a low sulfur diet, you could help this, but it hasn't been tested. So I can't say for sure if a low sulfur diet is going to work for ESO, but as time goes on, we're imagining that, assuming we don't have all these amazing drugs that we already are in development, which we can get rid of this SIBO, EMO, and ESO. I'm shooting for those. But if in the meantime we can't, there could be a vision of having 3 different diets, one for SIBO, one for EMO, low acid, low fermentables, and one for ESO, which would be low sulfur containing foods. But that needs to be developed, and we haven't done that yet. So there's a question about thoughts on the vagus nerve. Okay, so the vagus nerve is a nerve from your brain down the sides of the esophagus, past the stomach, and it controls almost everything up until the mid-colon. Controls, I mean partially controls, because the gut and the brain, if you cut the vagus nerve, you're not happy, things don't work well, you can get SIBO just from that. But you don't die. I mean, it's not like the gut doesn't function at all. So you can live without a vagus nerve, although it's miserable, but the vagus nerve is really important for functions. But what really strikes me and makes me think that all these years and decades of tricyclic antidepressants and treating the brain for IBSD is confused. And let me explain this. So the people who are very much into the brain-gut axis have done some really good work trying to understand the relationship between how the gut feels and how the brain responds, which is really incredible. But if you put somebody on an tricyclic antidepressant, you turn the vagus off. And this was the mainstay of treatment for IBSD for decades. And essentially you're slowing the gut down, which isn't what you would want in SIBO, and you're using the side effect of the drug more than anything else, because then they get constipated from the drug, and so your diarrhea is gone. But now there's vagal nerve stimulation. Well, vagal nerve stimulation turns on the vagus nerve, and now they're suggesting, that's evidence that it's a brain-gut axis issue. So I don't understand. You turn the vagus off, that's argument that it's a brain-gut axis issue. You turn the vagus on, that's argument that it's a brain-gut axis issue, and they both work. So it's a little confusing what to make of the vagus nerve, because if you turn it on or off, you're still making patients better, maybe. So, and we have some data coming out shortly that says maybe all of this is a bad idea, and I'll hope to share that with you in the coming weeks. And this is a study by Dr. Ali Rezai, which just got accepted today, but I can't talk about it yet. Is ESO strictly due to bacteria, or is it related to the metabolism of sulfur inside the gut cells in some way? So we've actually studied this. So your body produces hydrogen sulfide. Your cells produce a little bit, a tiny bit. And that hydrogen sulfide really is important in terms of smooth muscle function, nerve function. It has a lot of layers of function, but it also balances this special kind of chemistry in the cell called redox. And I'm not going to get into all the details of that. But when the gut is producing extraordinary amounts of hydrogen sulfide, we see the cells really crater. Their mitochondria stop functioning, the cells of the gut, that is. Their production of your own hydrogen sulfide goes to the boots because you don't need any more. You got way too much going on. And then you're basically, the cells start to die when you get to a certain amount of hydrogen sulfide, and in a process called ferroptosis. And so there's a lot of really bad things that happen with hydrogen sulfide when it's produced in the gut. So in answer to your question, hydrogen sulfide is okay, not bad, not good, terrible. So, and we see that with the breath test. So we did a study where we looked at the breath test, and the higher the hydrogen sulfide, the more destructive situation is going on in the small intestine. So there's a question here about COVID. So I've had this question a number of times, and I try to answer it with some care because COVID did change a lot of things. But my practice is super heavy on IBSD and super heavy on SIBO. We didn't see an increase after COVID. Now, we didn't study it objectively, but we really didn't see people saying, oh, the COVID made my SIBO worse or made my IBS worse. We didn't see that, although some other groups did and have published their results. So what does it mean to have hydrogen spikes after 90 minutes on the breath test? Somebody sent this question ahead of time. So hydrogen spikes after 90 minutes could mean a lot of things. So if you're on anything that slows the gut down, you can have a delayed spike in hydrogen, especially with lactulose. If you on a GLP-1, you could have a delayed spike in hydrogen. There are other reasons. There could be adhesions. There could be other reasons why the lactulose is taking time to go through. Or it's just normal. So we generally set the cutoff at 90 based on all the evidence, and every study we've done since that cutoff was set tells us that 90 is the ideal cutoff. Could there be some patients with SIBO above and below? Probably. You have to use that judgment as a physician. Aria sent a question. I just saw it. It just almost scrolled off. Is 4 times a year okay for rifaximin? So we looked at the use of rifaximin up to 6 times. And if it worked the first time, it almost always worked every time after that. This was a paper we published way back in 2011, I think. And so using rifaximin over and over again appears safe, and it's been used millions of times now since approval for IBSD, and doesn't appear to have any safety issues that have come up. In fact, it's so much more safe than tricyclic antidepressants, for example. But 4 times a year, I wouldn't approach it that way. If somebody was getting it 4 times a year, I would measure their vinculin antibodies. Are they positive? I would try to give them a prokinetic after the treatment with rifaximin to try to reduce the number of times you need rifaximin. A, because why do you need to take rifaximin over and over again? And B, it probably saves money and saves hassle because every time you relapse, you got to track your doctor down, you got to get another prescription or argue with your insurance. So, yeah, we try to reduce rifaximin use as much as possible. What do you think of Candida? I mentioned Candida earlier on. This is from Primark. I mentioned that it can occur. It usually occurs with SIBO, but it's usually quite a low percentage of patients with SIBO. Maybe 2 or 3% out of 100 might have candidal overgrowth. What about daily use of rifaximin? This is Lolly du jour. Daily use of rifaximin, I will tell you I do that in practice, but only for those whose vinculin is way over 3. And it's literally nothing is working and they feel great on rifaximin. And when I stop it, it comes back. I have some people in their 90s where, the motility really is struggling at that point for them. And they stay on the rifaximin daily and they do fantastic. The overgrowth won't come back. And that's, I use it that way. Is LDN, this is D. Tennant, 1982, is LDN a good prokinetic? LDN is used as a prokinetic. It also has anti-inflammatory properties. This is low-dose naltrexone, for those of you who don't know. But it's very modest as a prokinetic. Prucalopride or motegrity, the drug we use, is much stronger. Salvo Leila, hydrogen positive on breath test, vinculin positive with Klebsiella pneumonia and Citrobacter frundi overgrowth, chronic constipation, still rifaximin. So if you don't have methane and you have this overgrowth, I would treat it, and it's not methane, it's hydrogen, and you're sure Citrobacter is a hydrogen sulfide producer. So I'm worried about the Citrobacter causing hydrogen sulfide, and if you just give rifaximin, you may not get better. So 3-gas breath test is what I would do in this situation if you had the Citrobacter there, just to make sure how much hydrogen sulfide you have so we could track it. So I may not just use rifaximin. But if the hydrogen sulfide is not positive, I might give rifaximin. But if you're super constipated, and there's a question in my pre-questions, what about colonic inertia? So the patients so there's EMO, intestinal methanogen overgrowth, too much methane, constipated. But the constipation you get from EMO is very different than colonic inertia. Colonic inertia is no bowel movements for 10 days. Like, I'm talking zilch. If you have zilch, no bowel movement for 10 days, breath testing isn't the right thing. You will have overgrowth, but you will have probably hydrogen, but treating it is just going to make the bloating better. It's not going to make the constipation better. So colonic inertia is a different animal. So when you say chronic constipation, I would want to know the degree of that constipation, because if it's severe, then it could be overgrowth because of the constipation, not constipation because of the overgrowth, because they both can happen. So I hope that answers your question. One final thing is colonic inertia almost never happens in men. We don't know why. It's almost always, we're talking 99% of the time, it's female patients that would have colonic inertia. We don't see it in men. Somebody says EDS. What prevalence of SIBO emo has been reported or observed in your experience? Many of our EDS patients. So I can't give you an exact percentage, honestly, because we haven't looked back at that data in that way. But it's very common in EDS. EDS is Ehlers-Danlos syndrome, which can affect the flow of things through the gut because of changes in the connective tissue elasticity of the gut and other parts of your body, of course. And so the flow isn't good. And as a result, you get sagging of the bowel, and then that also contributes to worsening of flow and overgrowth. But treating the overgrowth and keeping it under control does help EDS patients symptomatically. And so it's important to do that. Okay, I'm going to take 2 or 3 more questions, and then we're going to have to wrap it up. But do you build, Aria is back to, you build resistance with too much rifaximin? So what we saw in the Target 3 trials with rifaximin, it was a requirement of the FDA to test for resistance. We didn't see any stable resistance as a consequence of 3 treatments of rifaximin in that trial. We didn't see any buildup of candida in stool from 3 treatments of rifaximin. So it probably doesn't cause candidal vaginitis or some of the other side effects of regular conventional antibiotics, as with rifaximin, it doesn't do that. So don't see resistance. So it works the first time, it works the 6th time. That's what we've published. Please, TXMKV, please tell us the average relapse time with antivinculin 2 without prokinetic. So if the antivinculin is elevated, what we see average relapse time would be about 2 to 3 months if the vinculin is elevated, on average, but it can be longer for some and shorter for others. But that's generally what we see. Do vascular compressions contribute to SIBO? Anything, so if you have a vascular compression, let's say you have an SMA syndrome, or you have a narrowing of the superior mesenteric arteries, so you're getting a little less oxygen to your small intestine. It's sort of like you crossing your leg too long, and then you try and stand on it, and the leg doesn't work. The small bowel doesn't work when it doesn't get enough oxygen, and it falls asleep, sort of like your leg falls asleep. And so you can get problems, and it can contribute to overgrowth potentially. AP Buddy, why probiotics seem to make symptoms worse? So this is I'm glad we end with this question, because this is a complicated answer, but the more we look, the more we don't like lactobacillus in the small intestine. I'm trying to clarify that. the colon, maybe that's where lactobacillus belongs, and you should have more lactobacillus, acidophilus, et cetera, but when you put it in the small bowel, it's a disruptor. It reduces the normal flora in the small intestine. And Dr. Mathur from our group has shown that in a scientific reports paper that unhealthy aging is associated with higher lactobacillus in the bowel. She's also shown that lactobacillus is associated with obesity. And we have some more recent data, which I can't share with you yet, from her work in the MAST program here, that lactobacillus has other bad properties that you'll hear about in the coming months. So unfortunately, lactobacillus isn't really great for the small bowel. So I want to thank you all. You guys did an amazing job. And, oh, yes, I just got past a note. So we have a new study that just came out, and we're going to be sharing the 3-gas breath test paper that just got published. There's going to be a video on our X feed and I think on Instagram Live as well. So you can see that paper because it really tells you how important hydrogen sulfide is and how dramatic it changes the gut, and maybe the more important of the 3 gases, to be honest, in terms of severity of symptoms. And I want to thank you all for joining me today. I know I didn't get to all of your questions, but man, they were great questions today. I really enjoyed this. I have to do this again, maybe after the DDW meeting, because at DDW, we have a lot of abstracts and some really new stuff. But if you are interested in the trial, I've already given you the phone number if you have IBSD and you've had a colonoscopy in the last 3 years. And so thank you so much, and look forward to helping you and talking to you some more on an upcoming IG Live. Thank you.